MSA is a really interesting case. So it's a rare disease. It's a very aggressive form of a Parkinson-like phenomenon. And we have a hallmark. We know that one of the things that happens when Abl is activated in the disease state is that it chemically modifies the synuclein aggregates. So we have a marker for it. But what's unique about MSA is that MSA occurs in a different cell type in the brain called an oligodendrocyte. It's a migratory cell. And so somehow the presence of the synuclein aggregates in the migratory cell gives rise to degeneration in the brainstem where Parkinsonism occurs. No one understands how that's occurring. So we first went and did the same thing that we did in the Parkinson's case. Could you take samples of post-mortem patient brain and look for the marker of Abl modification, that phosphorylation that occurs at a specific tyrosine, because that's what an Abelson Tyrosine Kinase does. And we saw that, we published that at the end of 2020 or early 2021. Then we started down the road of doing all the regulatory preparation for it. And so we hope sometime next year, probably realistically closer to the end of next year, well, we should initiate that trial. That trial's been in design for a while. We actually thought we might begin before the end of this year, and 6 months ago. It was clear it's a much more complicated case to set up the trial for that work. These are fragile patients. They are much rarer than Parkinson patients are. And so a lot more logistical work had to be done. And then, of course, we've had a terrible stock market in the US. We're fortunately public and cash flush, but I don't want to become not cash flush, not before I have some security. So we're also slowed the pace of the development of that trial work while we complete model studies, which we have that are not quite as sophisticated in MSA. Those studies have taken longer to execute, but we would like to see a therapeutic benefit despite the fact that we think Abl's involved. We'd like to see a therapeutic benefit in an animal model before we commit $10 million to a trial in humans. Again, that's just part of our philosophy. If we can preempt the effort in humans, knowing that something is unlikely to work, that's just being more responsible for shareholder dollars and a better use of our approach. On the flip side, if we continue to see the kind of things related to Abl that we think are relevant in diseases of this kind, it opens up the whole other realm. At least with ALS and with Alzheimer's disease, both of them are known to have an Abl component. It's been known for years in relatively crude Alzheimer's models that if you inject Abl inhibitors directly into the brain of an animal that you introduce the disease to, it's corrective to the defects that are seen. But we would never get involved in the Alzheimer morass, as I think we can all view it these days, before knowing that we really have an effective agent in human beings against the target. And it's much easier to measure that in Parkinson's because you're measuring people's ability to walk, or to put on a sock, or to brush their teeth. Those are things you can really quantify. Measuring cognition, how well people remember something, is exceedingly difficult to measure with a lot of noise. So if we have success in Parkinson's, we know going in the direction of Alzheimer's is going to be very beneficial, but we really have to think about how we execute on that. And that'll be benefited by the fact that some success in Parkinson's means I'll never worry about the financial stability of our company again. So, and that will be quite an aid if we could really have this kind of impact in an area of medicine that's had no luck.